We're now going to get into one of the main tools for life science or biology, and that's the microscope. And in this particular lesson, we're going to look at the history of some, and there's only a couple things that you need to know. Most of this you don't need to write down. And we're going to look at the different types. And your book talks about 2, and those are the only 2 that you're going to need to know. So as we go through this lesson, here's the key concepts. So make sure you're looking for these answers. How did microscopes change our ideas about living things? Because that's what we're talking about, classifying organisms. And what are the types of microscopes and how do they compare? The main vocab in the types of microscopes are the light microscope, the compound microscope, and electron microscope. The invention of microscopes enabled people to see details of living things that could not be seen with the unaided eye. So think about what we've just studied and how would this help? Now these you don't need to write down. I'll tell you when we get to the right one. But long before Christ, in 1000 AD, the first vision aid was invented. They don't know who. It was called a reading stone. And it was a glass sphere that magnified when laid on top of reading materials, as you see in the picture. Then these were the first pair of eyeglasses. And I'm not sure exactly how they wore them. But that again used lenses, which is magnifying. 2 Dutch eyeglass makers, the Jansens, experimented with multiple lenses in a tube, and so they actually are the forerunner of the compound microscope and also the telescope because of their work with the lenses in the tube. Now here's one you do need to write down, and we're going to study more about Robert Hooke and his discoveries about the cell. But in 1665 he published an article, actually a little booklet, and in it Robert Hooke had his drawings and also wrote some things about what he saw under his microscope. Now, his microscope only magnified about 30 times, and he was looking at cork under it. And he called a cell a cell, so that's something else you need to know. Robert Hooke is actually the one who named cells. And he named them because when he saw them under the microscope, they reminded him of cells in a monastery and cells in a prison. So he called them a cell. Then another guy that you need to know about microscope history is Anton van Leeuwenhoek. Now he built a simple microscope, had only one lens, so it wasn't a compound microscope. He looked at all kinds of stuff, and you can see some of them listed there. And he also came up with methods for grinding and polishing the microscope lenses, which allowed then a change in that lens to take some curvatures, which enabled it to magnify more times. So you need to know in the 1600s, Van Leeuwenhoek had a microscope, and it could magnify 270. Now remember, in 1665, Robert Hooke only magnified 30 times, so that was a big increase from 30 times to 270 times. In the 18th century, they had all kinds of technical innovations that improved microscopes, leading to microscopy, which becoming the popular among scientists back then, and lenses combining 2 types of glass reduced what they call the chromatic effect, and that's a blurriness, especially around the edges, and these halos. They also call that about the moon. There's a chromatic effect too, the chromosphere, and that was this type of lenses then was trying to reduce that so that you could see the image more clearly. 1830, Joseph Leicester, he reduced the chromatic effect, and he used several weak lenses, and it gave a real good magnification without blurring, and so it actually is like the forerunner of a compound microscope. It became that. And then we have Abby, or AB, I'm not sure how you say that. He came up with formulas to help with the resolution of the microscope. The resolution has to do with how crystal clear you can get the image. And then we have, I'm not sure of these names, Zsigmondy, and he developed that scope right there called the Ultra Microscope. And he actually won a Nobel Prize in chemistry for it. None of these you need to write down, so don't worry about them. Then Zernicke, he invented a phase contrast microscope, and that allowed the study of colorless and transparent biological materials, and he too won a Nobel Prize. In 1931, we had this Ruska, who invented an electron microscope, and he won a Nobel Prize in 1986 for it. Now, electron microscope is very different from light microscopes. See, light microscopes are using lenses and light to magnify. The light is going through the specimen. An electron microscope is using electrons instead of light. And here's his first electron microscope. Notice electron microscopes, they can view objects as small as the diameter of one atom. And then these two guys came up with a scanning tunneling microscope, gave three-dimensional objects, and another Nobel Prize winner. Now the types of microscopes that you need to be familiar with are the two that we will be using, the compound microscope, sometimes referred to as the compound light microscope, and the dissecting microscope, because we'll be using both of those. And that's the image on the left there is a compound microscope. The one in the middle is a dissection microscope. And then the electron microscopes, there's only two that you need to be familiar with that your book talks about, the scanning electron and the transmission electron microscope, SEM and TEM. And there on the right is a diagram of the electron microscope. So compound microscope is a light microscope. It can magnify up to 1,500 times the original size of the object that is put on the glass slide. Our total magnification, so the highest that our microscopes will magnify, is 1,000 times. So they're pretty strong compound light microscopes, whereas the best ones can go up to about 1,500. Compound microscopes are light illuminated. They used to use mirrors way back. Now we have actually electrical lights, and we actually have cool electrical lights, which is much better for seeing and not get heating up and so forth. And they give us pretty good high magnification, but low resolution. And low resolution means it's not going to be as crystal clear as when you get to the electron microscopes. Here's some images taken with a compound microscope. So notice the difference there. The blood 1,000 times, the piece of wood or part of a wood 200 times. Dissection microscope, and there's a picture of one instead of a diagram. They use light as well. You can really get some neat images with this because it has different lighting on it, and you can change the lighting, and you're going to get a three-dimensional picture, whereas you only get two-dimensional with the compound light microscopes. And you can't look at individual cells because it's not like the light is passing through a real thin specimen, but it's going to enlarge and let you see like inside the parts of a flower or inside of something you're dissecting, like the worm. It's also called a stereo microscope. Here's some images taken with a dissection microscope, and that's the head of a moth pupa at 60 times. And then in the upper right-hand corner it's a sunflower cross-section, and they got the arrow pointing to a pupa, which is in the stem of it. And that's only 10 times. Now, ours do not magnify up to 60 times. Now, here's the biggies. We don't have those. I don't think any high school would have one of these. Only some colleges have them because they're very expensive, and they also have a high cost of maintenance to them. The two that your book mentions are the first two, the transmission electron microscope and the scanning electron microscope. Some other ones are called reflection electron microscopes and scanning transmission electron microscopes and low voltage electron microscopes. And there's a variety down there, and you can see how massive they are. The one you need to be familiar with, the scanning, the SEM, it gives you 3D as well, but it gives you very high magnification along with high resolution. And it says it coats the specimen in gold, and the electrons bounce off and give you an exterior view of the specimen. The pictures are in black and white. Now they can actually change them into color, but it's not being seen through the microscope as color. And there's a picture of one. Here's some images. Now look at the detail in the antenna of a cockroach and the pigeon's blood. Those are actually taken with a scanning electron microscope. A transmission electron microscope, TEM, it only gives you a 2D view, and you have to slice the specimen real thin, and the electron beams pass through it, and it too gives high magnification, high resolution. I've read some places where when they get a picture from on the highest power of an electron microscope, then they actually can enlarge the picture, I think like 5 times, which can give a magnification of up to like 5 million when you blow up the picture. Here's some pictures taken with an electron microscope. The transmission one, TEM. Okay, using microscopes. They have a lot of uses. Of course, you know that doctors use them. And then also they use them for surgery. When they have to do surgery like in the brain, where it's very delicate in areas where they need to be able to see the actual nerves, they're going to use microscopes to do that. The word microscope came from Latin, as many of our science words. Microscopium, I guess is how you say that. And it means an instrument for viewing what is small. So that's why it's magnifying. Using microscopes, there are many uses. Forensic scientists use them all the time to study evidence from crime scenes. They also have the comparison microscopes, like for ballistics, to match up the striations on bullets. People who study fossils use microscopes to examine the fossils and other materials from where the fossils are found. So they're used by all different kinds of sciences. Now here's the big idea from your book. All living things have certain characteristics in common and can be classified using several methods. The invention of the microscope has enabled us to explore life further, which has led to changes in classification. Now remember I said there's a committee that meets, and sometimes they learn more information about an organism and they have to change like its scientific name, or they have to change maybe what phyla or subphylum it is in. Well, here's why. The better microscopes, the more magnification and resolution, then we find out more things about those organisms. And we'll talk about how to use the microscope and care for it when we start going into the lab to look at cells under the microscope.